Okay, let's get started. Hopefully everyone is settling in comfortably into the Zoom room. Welcome everyone to this fourth webinar in the IMG webinar series. My name is Reddy and I am part of the Prokaryotic Super Program here at JGI. I manage Genome's online database. Today you are going to learn about data submission and management in IMG. Our goal is to give you an overview of defining projects in GOLD, submitting your sequencing data to IMG for annotation, and managing your submissions. Some of you attended our previous webinars on Genome Search, Advanced Search Builder, IMG Data Export and Download, and the last one was on metagenome bins. Today's presentation and live demos will help you to understand how the data shown in those webinars, as well as other data you use in the IMG system, comes into existence. In other words, the steps it goes through before you see the data on IMG and use it. So those are the things you will be learning today. If you couldn't attend those webinars, no problem. Recordings are available on IMG website. You can watch those at your convenience. We aim to complete this webinar today in 60 minutes. If we go over a few minutes and if you have to leave, don't worry. A recording of this will be posted on the IMG very soon. During this webinar, you may use the chat window to ask questions. They will be answered immediately or posed to the speaker by panelists. Our answers will be provided in the transcript later to you, all users, attendees. Thank you for your attention, and now I will move on to the first part of this presentation. Today, I'm going to talk about Genome's online database and how to define an isolated genome project in GOLD for its annotation in the IMG system. Before I dive into more details about GOLD, I want to give you a brief overview of the Giant Genome Institute, where GOLD and IMG databases are hosted. JGI came into existence in 1998 and is part of the Lawrence Berkeley National Laboratory. Around 280 people work here. We are funded by US Department of Energy's Office of Science. We are a DOE user facility. You may wonder what a user facility is. DOE user facilities enable large-scale sciences that are not generally possible to carry out in individual labs or core facilities at your universities or organizations. So the community science program calls every year, JGI invites proposals from research community in DOE mission relevant areas such as bioenergy, carbon cycling, and biogeochemistry. Using the state-of-the-art DNA sequencers, advanced genomic technology, and the computational infrastructure here at JGI carries out metagenomes, plant, fungal, and microbial sequencing programs at a scale which is generally not possible for individual labs in their universities or using their core facilities. But only this is kind of stuff is facilitated by the user facilities like JGI. This is the big picture of GOLD and IMG systems from a user perspective. Imagine this is you coming here to use IMG and GOLD databases. A user can access GOLD's login-free public site or use a system that requires the login and the curation interface to enter your projects or come back and later update the projects you already entered before. On the other hand, you can access IMG system and try using the favorite tools are the ones you were shown in the previous webinars. You can directly access that from the IMG system. If you want to submit your own projects to IMG for annotation, you will first come to the GOLD and define those projects here in the GOLD database. Once you do this, these projects will be available in the IMG MER system for you to upload the sequence data and submit. And the projects you entered in the GOLD are available in the IMG submission system as soon as you create them here in the GOLD. Once you submit the projects, you can come to the submission system to check on the status of your submission. So usually these bidirectional arrows indicate the back and forth of the transfer of the data and the information between you, the user of the GOLD database, the GOLD and the IMG submission system. In a nutshell, this is how you interact with GOLD and IMG systems as you define your projects and submit your sequencing data for annotation and manage those submissions. So GOLD acts as a gatekeeper to the IMG system for defining projects and managing metadata to facilitate their annotation in the IMG. And the genome portals are one-stop shop for providing unified access to all JGI datasets. You can download both the raw and analyzed data using the genome portal. Throughout GOLD database and in the IMG system, you will find links to the portal pages. So what is GOLD? The Genomes Online Database is a centralized resource for cataloging sequencing projects from around the world along with their metadata. So what is in GOLD is basically what I just described. We host a list of genome and metagenome projects along with the list of metadata. What is not in GOLD? GOLD doesn't hold any host any sequencing data. Sequencing data, their annotations and comparative analysis tools are hosted in the IMG system. What are the different sources for the projects in GOLD, or how these projects are coming into GOLD? So projects carried out here at JGI as part of the community science program are one of the major sources. If you are the lucky few to get your proposals accepted by JGI and have your project sequenced here, those project details are imported into GOLD from our JGI system automatically. So there is no need for the PIs or the PIs labs to enter those projects into GOLD. Individual users from around the world, the second stream of projects into GOLD are from the individual users. They can enter their projects straight into the GOLD. If you have many projects to enter, we can provide you a batch load option. You need to first enter at least a couple of projects on the GOLD website and then contact us and we will provide a batch load Excel. We take this approach to let users experience the project entry interface and get feedback on their entries before they spend more time on assembling large batch load Excels. And the third stream of projects into GOLD are from public resource repositories like NCBI. So these are the three major sources for the projects you see in GOLD system. This is an overview of the GOLD database. In GOLD, we use a 4-level classification system to organize different entities. At the very top, we have study. Under it, we have biosample or organisms at the same level, and sequencing project and analysis projects are at the 3rd and 4th level. At the very top, the study denotes the overall aim of the research you are undertaking. Say you are setting out to sequence soil samples isolated from a forest throughout the year to study seasonal changes in the microbial diversity. Or say you are setting out to sequence few bacteria that are plant pathogenic bacteria. So the details corresponding to these kind of research initiatives are captured here at the study level. Under the study, we have biosample or organism entities. Biosamples represent the environmental samples you collect from the field or from a subject in case of host-associated metagenomes, either it is a plant or an animal. And the organism entity is used to define individual living things like plants, fungi, bacteria, or viruses. And this also includes the single cell organisms. And at the third level, the sequencing project entities define the sequencing work carried out for a given biosample or organism. A biosample can be used in a metagenome or metatranscriptome project. Cultured isolate organisms or cultured single cells organisms can be the basis for isolate whole genome sequencing project or a single cell whole genome sequencing project. At the fourth level, you see the analysis projects. They represent a set of computational and informatic processes applied on the given sequencing project's output, which is the sequence data. They will be specific to sequence types, as you see in the linear paths here, like the study by sample, sequencing project, analysis project. So for example, if you sequenced an isolate whole genome sequencing project, the analysis project you create on it would be isolate genome analysis project. And also you will see here the combined, some special analysis projects, which are grouped here that takes multiple sequencing projects. So these are combined analysis projects. So you may combine sequence from two metagenomes if they are from similar locations, and you think that by combining those, you may get a better assembly. So in that case, you can actually combine two metagenome projects of those are the same type, and you can have a metagenome combined assembly. In another example, say you sequenced a single cell genome project, and you also happen to have a metagenome from the same environment, and you thought that, okay, by using the sequence from the metagenome, you can get a better assembly of the single cell genome. So in that case, you may combine the sequence data from metagenome and the single cell, and you can get a better assembly. So these are the four different levels in the GOLD, and this is how they are organized and they are interconnected. And each of these entities, they have different IDs. They start with the prefixes of G, S, GB, like that. The G stands for GOLD, and the S, B, they stand for either study, biosample, organism, project, or analysis projects. In today's demo, I'm going to show how to enter a study, organism, an isolate sequencing project, and the analysis project. If there are any questions so far, I will take a moment here to answer those before I proceed. Okay. Looks like there are not any questions, so I will proceed. So far you heard a few times about the metadata. Why do we need to care about the metadata? Is it because GOLD curates and requires you to provide the metadata? No. In the previous IMG webinars, you have seen in action how GOLD's metadata is used and driven advanced searches. For example, when you try to look for samples or genomes from a given set of environments, it is the GOLD metadata that enables those searches. For performing meaningful comparative analysis tasks, you need to know more about the samples and organisms' metadata. Without metadata, it will look something like this. So if you have a map with no notations on it, or if you have a supermarket where you can find cans with no labels on it, can you imagine making use of this map to go to an address or shopping in a store with such a merchandise display? I'm sure this will be a challenge for most of us, and that's how challenging it is going to be to use scientific data without metadata. How about this? Having an annotated map and also a set of neatly labeled cans in a supermarket, this will definitely help you to find an address you are looking for, or dramatically enhance your shopping experience, right? In the same way, scientific data with well-annotated metadata is more desirable and help in promoting a more meaningful comparative analysis tasks. So this is the GOLD help page I want to bring your attention to, which you can use while trying to enter the projects into GOLD. There are several links here, the GOLD documentation, and contact us with feedback or questions. If you ever have any questions or you want to submit feedback, you can use this form. And also we have a link to the GOLD terminology where all the terms used in the GOLD are listed. And also we have this frequently asked questions. Feel free to use the chat window to ask questions, and we may include some of these questions if they are of general interest in our in future here. And also I listed here in the presentation some useful links that you will use if you when trying to enter a project into GOLD. And also I listed the example plant pathogenic live demo today and the corresponding bioproject. Yeah. These are all the wonderful people behind the GOLD database. Nikos is the head of the prokaryotic super program. Amy oversees both IMG and the GOLD systems. Myself, Reddy, and I manage the GOLD database. Dimitri and John are our software engineers. Supratim, Jagdish, Galina, and Mahati, they help us in the data curation. So I will stop here and now switch to the web page for the live demo. Bear with me for a few seconds while I switch. Okay, so this is the GOLD homepage. In order to enter projects into GOLD, you need to first log in. Right now, I did not log in, so you got the instructions in the email how to obtain a JCI single sign-on account. You will be using that to log in into GOLD. So now I'm logged into GOLD, and I want to show you, bring your attention to a few things here. This is the help page I pointed in the presentation. So here you have the links to GOLD documentation, GOLD terminology, and the frequently asked questions. This GOLD documentation has a project entry help document. This is a PDF with, it's a long document, but this is going to be very informative for people who are trying to enter projects into GOLD. You will find screenshots, you'll find these forms, filled forms with the screenshots. So this will basically step by step, it will help you to go through the process of entering projects into GOLD. And also we have the GOLD terminologies here, where different terms used in the GOLD, they were defined here. And another thing I want to point out before start entering the project are this NCBI import tracker, where we import projects from the public repositories like NCBI. Here you see all the NCBI prokaryotic projects and the projects that we so far curated and imported into GOLD. And this bar indicates the number of projects that were already annotated and available in IMG. So if you ever interested in a public project, you can always check to see if we have imported into GOLD or if it is already in IMG before you make an attempt to enter those projects. So before, how you define projects in GOLD? You come here and click the register link here. And here, create a new sequencing project in GOLD is the link that you are going to use it. So I want to point out here, there is a link here to the GOLD project entry help document, the one you saw a few seconds ago. And also if you have a metagenomes, and we follow a special metagenome naming convention, and there is a link to the document, metagenome naming conventions document here. So I'm going to start now with entering a sequencing project. Before I start, I want to point out since I'm taking a publicly available genome sequencing project, I want to point you to the NCBI bioproject page where the project information is available. So this is a NCBI bioproject page, and this is the project I'm using today in the entering into the GOLD database. So I want to point out to a few different accession IDs here. This is the NCBI bioproject accession. This is the bioproject ID. And the organism that is described here is this Clavibacter michiganensis, and the NCBI taxonomy ID is this. So all these are the different IDs that are available. Often users get confused, so I just want to point it out. And also we have here NCBI assembly accession and the GenBank IDs. Okay, I'm coming back to the GOLD project entry page, and I'm clicking on the sequencing project. So I see select the type of submission you wish to make. So here we have two options. One is biome and another is the organism. So I just mentioned I'm going to show how to enter an isolate. So we are going to choose the organism. But you may be wondering what is biome. So you can always refer to the GOLD help pages when you have such doubts or questions, and you will find all the terms described there. Okay, so here I checked GOLD organism. Now, if you, now I am taken here to this page to enter the GOLD study, that the very at the top level. So if you are a returning user and if you have a study already entered into the GOLD, you can actually search here and use that. Otherwise, you will be entering a new study here. So for today's demonstration, I'm going to enter a new study here. My study name is, I'm going to enter the study name as the comparative analysis of wheat pathogenic microorganisms. So other names, if your study have any other names, you will enter it here. Otherwise, you leave it blank. NCBI umbrella bio project. So this applies to some bigger projects, for example, like human microbiome project. So that involves like hundreds of microorganisms, but all these things were grouped under another bio project that is called umbrella bio project. So if studies that consist of those umbrella projects, we have a umbrella bio project to enter here. Otherwise, most of the time this will be empty. And the PI name, I'm entering my name here. And the email, here I want to point out to most of the users, when you are entering, try to enter your PI name instead of your name, because once your postdoc or graduate studies are over, you may move on. But then PIs, they will always come back to us asking to update the information. But you, as the person who are entering, your name will be recorded as added by in the system, and you will have the full access. So there is no problem. You can always access your entries. So just to make things clear, always try to enter your PI names instead of your own names. And the description here, you briefly describe what the study is about. So I'm just entering the organism name here, that the first organism that I'm going to add under this study. So the relevance. So here you can select one or more relevance terms from the controlled vocabulary list we have. So here, say I'm choosing comparative analysis, and I want to add one more term. That is, I know this is a plant pathogen, so I want to use that. And then ecosystem classification. We know this is a plant pathogen, so this will be host associated. Plants, and let's say this was isolated from a leaf. And we don't know beyond that, so we will leave the as unclassified. And now I click on the create new study button. Okay, so a new study is created, and yes, I'm copying the organism name from here. So here we have two choices. One is add a new organism into GOLD, and another is search for an existing organism. So in case of organisms, most likely you don't need to click this form and enter a new organism, because we already imported all the public organisms from NCBI system and from other resources, and they are available in the GOLD system for you to choose. Here you see like organisms total available around 400,000. So the chances of you entering a new organisms are low. So here what I'm doing is I just copying and pasting the organism name I copied here. So it takes a couple of seconds to search for the organism. If you have more organisms, like this, matching your genus species, you see you get a long list. It may be difficult to search. So in that case, what you can do is you can come back here and you can use the specific identifier to search. See, now miraculously, you just found one organism that is very specific, and you can just select without wading through a long list. So you selected an organism, and then you are click next to proceed to creating a sequencing process. So what did we do so far? So far, we created a study and we selected the organism, and now we are at the process of creating a sequencing project. So the sequencing project name is already populated for you here. And there is no, if you have any other name, you will enter it here. Otherwise, you leave that thing blank. NCBI bioproject name, since we are taking this from the NCBI system, we are entering the NCBI bioproject name and the bioproject ID. These are all everything that we saw here. NCBI bioproject accession, NCBI biosample accession. So I'm going to leave this field empty. The reason is we already have this project in the GOLD system, and we don't want to allow people to create duplicate projects. So we have some constraints on this. If I enter the real biosample here, it will prevent us from entering this project. So I'm leaving this thing blank, the locus tag, and the sequencing center. All the fields that you see with this asterisk are the required fields. So we always need to provide a sequencing center. So I'm going to choose my grad school here. And the collaborating institute. Say I'm collaborating with the JGI and the project and the funding agency. Say you got your funding from NSF. You will choose the funding agency there. And you will provide a project description. So here I'm providing a description saying I'm out to sequence the Clavibacter bacterium. And this is not a test project. This particular field is only available for admins. You may not see. And the specimen is already preset to organism. And we are selecting DNA because this is a genome sequencing project. And the sequencing strategy is the whole genome sequencing. So the sequencing status, say you already, if it is in progress, you will set it in progress. I'm setting this complete. And the sequencing quality, standard draft, high quality draft, there are 5 different things to choose from. And the finishing goal, say here you are set out to generate a finished sequence, but the one you have now is the high quality draft. So the sequencing current sequencing quality is the high quality draft, but your goal is the finish. So I'm setting this as finished just to show the difference. Yeah, the sequencing technology used, say you are using PacBio and the library method, number of reads. So and if you know the chromosome count and the plasmid count, you can enter those details there. So I'm just entering some number here. And now create sequencing project. Okay, so far what we did is we created a study, chosen an organism, and we created a sequencing project. And next step is to create an analysis project. So since we are doing it in one go, the sequencing project we just created is already pre-populated here. And both the project name and the study names were populated for you here. And I'm entering, say, this is a reassembly using additional sequencing. Since this is a project that already existed, say you did some additional sequencing in your lab, so you're coming here to create another analysis project. So basically the details of the analysis project that you enter here. Non-SP owner AP. So what this means is users who don't own sequencing projects, that means you can come to Gold, choose any public sequencing project, and you can create an AP on it. That's not the case here, so that's why I'm setting this to no. And this is no, this is the IMG use is standard. Again, this is a admin field. You won't be able to see online when you are trying to create an AP. Assembly method, this is a required field, and also you need to provide in the specific format like you see here with the version number. Whenever you have questions or doubt about what to enter, and if you see this tooltip, you can mouse over it to see with examples how the value is going to look. And the PI name and the email are already pre-populated here. And external references. So I'm just going to enter the GenBank ID and the assembly accession here and submit. So now we created an analysis project. So we went through the whole process of creating a study, choosing an organism, creating a sequencing project, and an analysis project. So the analysis project, just the one we just created, is GA0438804. This is the ID you will need to go to the next step of submitting the data to IMG for annotation. So at this step, you can click on the link and go to IMG submission site to submit the data. Or if you are busy or you got distracted at this step, you can always come back and submit the data to IMG. So if you have any questions, I will take now. Otherwise, I will stop here and hand over this analysis project ID GA0438804 to Marcel to show how to submit data to using IMG submission system.
Reddy, there was one question which was asking about the sequencing quality. How do you determine the sequence quality of your project? You had the pull down where it said finished draft, finished or improved draft. How do you go about determining that for your submission?
Yeah, that's basically like when you are, these are the 5 different levels of the sequencing quality described in a publication we have in our help doc in the terminology to link to that descriptions. In generally, like if you have the sequence, if it is with a lot of gaps, and if it's that it is only contigs, if there are many contigs, usually that's not a finished quality. So that would be a low, like a standard draft or high quality, things like that. So it's basically, it is the set of criteria you apply. Is it at how many contigs are there, or if there are more less number of contigs, and it is without any gaps, that will be the finished thing at the higher level.
Okay, so it's partially subjective, right?
Yes, yeah, I think yes, yeah. There is no, it's not like any metric to, yeah. It's you're right. It's a subjective. Yeah.
Okay, thank you.
Okay, I'm stop sharing the screen and I hand it over to Marcel.
Okay, thank you, Reddy, and hello everyone. My name is Marcel Huntemann. I'm a software developer in the IMG group here at the JGI, and I would like to continue the process that Reddy already started and show you how to submit your own data set to IMG for annotation, how you can track its status, and also how to share it with your colleagues. That part is going to be a live demo. And additionally, I will also present a couple of slides listing the tools and databases we are using in the annotation pipeline that gets executed on your submitted data. And after that, I will briefly go back to IMG for a quick look at an example of how a fully annotated dataset looks like once it got loaded into the system. All right, let's get started with the dataset submission then. As already mentioned, you have the possibility to go to IMG's submission site directly from GOLD after you created your analysis project ID, or you could come back to it at a later point. In that case, the easiest way to access the submission site is to go to the IMG homepage, and here either go to the IMGM menu and click on Submit Dataset at the bottom, or just use the Submit Your Data button that's right on the landing page. To submit your own data, you need to have a JGI single sign-on account. And I know that everyone that signed up for this webinar already got the information on how to create one. But for the people who are watching this video later, I quickly want to point out that you can create a JGI single sign-on account by simply clicking on Register and then filling out the form. All right, so I'm going to log into my account now. And once it logged us in, we'll land on the IMG submission homepage. And to submit a new data set to IMG, we want to click on the New Submission tab here at the top of the page. Now, the first thing it's going to ask us for is in GOLD Analysis Project ID. And conveniently, Reddy just showed us the exact steps necessary to create one of these. So I'm going to enter the AP ID for which I want to submit a data set here now, and I will use the exact same one that Reddy just created. And after that, I just click on Submit Dataset to IMG. And the submission site then connects to GOLD and pulls out some basic information like project name and type so that I can double check and make sure I got the correct project. This looks all right to me. It's the same Clavia-Bacter organism I want to submit the sequences for. So I'm going to click on Submit Analysis Project. And this will then prompt the IMG submission agreement, and I encourage everyone to actually read it before just agreeing to it. It's short enough, and most of its information was actually already displayed on the submission site's homepage. It basically lists which metadata we consider to be mandatory and says that if any of it is missing, we are going to cancel the submission. Then it says that we keep isolates private for up to 18 months and metagenomes for up to 24 months. And after that, they automatically become public. And it also states that you can't get around this by deleting the data set shortly before this period ends. It mentions that multiple versions of the same genome or metagenome will be kept in the system for up to 6 months, after which we remove all but the latest version. And at the end says, as Reddy already mentioned earlier, that all the assembled and annotated data can get downloaded via JGI's genome portal, and that we are not depositing the data into GenBank for you. All right, so I read it, think that it all sounds reasonable, and then I click on I agree. And this now brings us to the actual submission form. And as you saw from the displayed analysis project information 2 screens back, I'm trying to submit a Clavibacter isolate genome here. But this screen would look exactly the same if I would submit a metagenome instead. You would also have a submission information tab and a submit sequence file tab with the exact same fields, just the pre-filled project data would be different, obviously. by default, the visibility of all newly submitted project is set to private so that only the submitter can look at the data. If you want to make your data set public in IMG from the get-go though, you can do so via switching the is public in IMG database value to yes. In my case, I want to keep it private, so I'm going to leave it at no. The next value I can change is the sequencing status. The options are draft, permanent draft, and finished. For metagenomes, only draft and permanent draft makes sense, obviously. But if you submit an isolate, you might actually have a finished version for which all gaps have been closed. This is exactly the case for the genome I want to submit, so I'm going to change that value to finished here. And the last field on this tab can get used to replace an already existing data set in IMG. Every data set that gets loaded into IMG gets assigned a 10-digit numeric identifier called IMG taxon OID. And let's assume a while back you submitted a first draft version of your genome or metagenome, and you now have a better version of it, or maybe even a finished version in case of an isolate. And you would like to use the new one in IMG instead of your previously submitted draft version. In that case, you would put into this replace taxon OID field the IMG taxon OID of your previously submitted draft dataset. And by doing so, this new submission will automatically replace your draft dataset when it gets loaded into IMG. Also, if at some point you are not quite sure about the meaning of any of these fields, you can always click on these tooltip icons and a little help text with an explanation will pop up. Okay, so I don't have a data set in IMG that I want to replace, so I'm going to leave this field empty and we'll switch to the Submit Sequence File tab now to provide the actual sequence data. And the first thing I would like to mention here is that the only format IMG accepts sequences in is the FASTA format. So no FASTQ, meaning we do not accept reads and do the assembly for you. The data that gets submitted must be assembled. If you're not sure about what the FASTA format looks like, I encourage you to watch the recording of our IMG data export and download webinar, in which Natalia showed examples of the different file types and formats IMG provides via the earlier mentioned Genome portal, one of which is the FASTA format. Another thing I'd like to point out is that if you gzip or zip your sequence file to compress its size, please make sure that the compressed file really only contains the one FASTA file you want to submit. I've seen cases in which the uncompressed files contained either folders or multiple files. The pipeline attempts to decompress the compressed file directly into a single FASTA file though, and this operation obviously fails if there are multiple entries in the submitted file. Okay, so I have a assembled FASTA file on my hard drive here that I want to submit, and thus I'm going to click on the Choose File button here. This brings up a window that lets me select the file from a hard drive. And in this example, the upload is very quick. But if you want to submit a big metagenome of several hundred megabytes or even a couple of gigabytes, you probably don't want to sit in front of your computer and hope that the upload finishes before the browser times out. For those cases, we also give you the option to provide an URL in this field below instead. You can put your compressed or uncompressed FASTA file on a FTP server or a public website and paste the URL to it in here. The pipeline will then do a wget on the URL and download it automatically when it starts processing your submission. The link must be publicly available though, meaning it cannot require any kind of login credentials. And again, another word of caution here, Google Drive links unfortunately do not work, since those links actually point to a website that then displays the file or a download button. The provided URL really must point directly to the file that you otherwise would have selected via the Choose File dialog. The next field provides us the option to also submit a coverage file. Coverage files are one of the standard output files of assemblers that list each created contig and how often it is represented in the dataset. These files usually get submitted with metagenomic datasets and are used by some abundance-based tools in IMG. And again, I have the option here to either upload it from a hard drive or provide it via an URL. The last field lets me set the topology of my sequences. For metagenomes, this will obviously always be linear. But for some finished genome, all the sequences usually correspond to circular replicons, which they do in my case here. So I'm going to send the topology to circular. Well, and this is already all that's needed for a dataset submission. After this, I'm going to click on Submit. And on the next page, it's going to tell me that a new submission got successfully created and what its IMG submission ID is. If you have any questions in regards to your data set before it gets loaded into IMG, this is the ID you want to provide to us in your correspondence, since we can use it to look up your data set when it doesn't have an IMG taxon ID assigned to it yet. Right. Clicking OK brings us to our personal submission status page. And this page now lists all of my submissions and their current states. The two fields to check on in the beginning are approval status and status. Approval status currently lists pending review. This means that somebody first has to check that all the mandatory metadata required is really present in GOLD and that it also makes sense. If that's the case, the person will switch this field to approved. And only when the submission is actually approved, the annotation pipeline will go and pick it up. Once it did so, you will see the status field changing. It will first switch to annotation in progress, then to annotation done, pending for loading, then data loading in progress, and finally to finished. Unless, of course, there's an error at some stage. In that case, the status field will say that a particular step failed. And when you click on your submission ID, the following submission details page will additionally also display a comment field in this area here that usually lists what caused this error. It could be that something on our side failed, for example, the compute node which processed the submission on the supercomputer crashed, or there were some issues with its file system. In those cases, we would reset and reprocess your submission after those issues are resolved. In other cases, it could be due to some issues with the data you submitted, like the multiple entries in the submitted compressed file issue I mentioned earlier. And in such a case, we would actually cancel your submission and you would have to resubmit a corrected file via new submission. All right. Back to the submission page. Once the status field here says finished, You will also see a 10-digit number in this IMG taxon OID field, which now identifies the data set in IMG. By that time, you should have also received an email stating that your data set got loaded and how you can access it. As mentioned earlier, by default, your data set is private and only visible to you. If you want to share it with a specific set of people, though, while still keeping it private, you can do so by listing their JGI single sign-on usernames here in the Grant Access To field. Now, let's assume that Reddy and I work together on this project, and I want him to have access to the data set as well. Then I first select the data set I want to share with him. And afterwards, just type his username into this field. And if I have more people I want to share it with, I can add their usernames here separated by commas. And after I click on the Grant Access button, it tells us that user T.B. Reddy was granted access to our submission. So now Reddy will be able to see the data in IMG once it got annotated and loaded. All right, and last but not least, in regards to the submission process, I want to point out, since you probably don't want to sit in front of your screen the whole time and wait until the status fields actually change, that you can always come back to your submission status page later. For that, you just log in to the submission homepage again, like we did in the beginning, and then click on the Submitted Datasets tab here at the top of the page. And this will bring you right back to it. All right, and with that, I will now switch to the slides. To give you a brief overview of which tools and databases the annotation pipeline uses. Since I assume that most of you would like to know how we actually process your submitted data set and get to the results displayed in IMG. So the annotation pipeline has 4 main steps. First, we do some data pre-processing. Then comes the feature prediction, which for isolate is followed by a QA/QC step. And in the end, we are doing the function annotation, and I will now go over each of these steps and show you what happens during them. So before we start with the actual gene calling, we do a little QA on the submitted FASTA file. We develop these steps based on the submitted data we encountered over the years, which in rare cases cause some of the used tools and with that the whole pipeline to fail. To avoid that, we first remove all empty lines, carriage returns, and white spaces before the sequence names in the file. Then we make sure that not most of the sequence data are non-ACGT letters. If that is the case, though, the submission would automatically get canceled. This would also happen for isolate submissions that contain more than 500 sequences per million base pairs. And additionally, we remove all sequences shorter than 150 base pairs from the file, and then replace all non-ACGT sequence letters with ends. In case there are sequences afterwards that only have ends left, we also remove those, of course. And then at the end, we make sure that there are no duplicate sequence names in the file and that they are long enough so every tool can handle them. For that, we are renaming the final sequences with the GOLD AP ID, followed by an underscore and a sequence number. We know that some people already start to analyze their submitted data while they are waiting for it to get annotated, and then likely want to compare some of their findings to the ones displayed in IMG later. So for those cases, we provide a mapping file that maps the sequence names as listed in the originally submitted files to the sequence names as they appear in IMG now. All right, after these pre-processing steps, we have a final FASTA file that we then use as input into the feature prediction phase. And here we first try to find the tRNAs via tRNA scan SE. Then we predict the rRNAs and other non-coding RNAs by running CM search against the RFAM database, followed by the CRISPR prediction via a modified version of CRT. Then we run Prodigal and GeneMark to find the CDS genes, and in the end, take the results from each of these modules, combine them, and make sure that there are no conflicting overlaps left afterwards. So after this step, we have a final GFF file containing all the predicted features, and an additional FASTA file containing the protein sequences of the CDS genes, which we then use as input for the functional annotation phase. But before we do that though, for isolates, we first run another QA/QC step to make sure that we don't load any bad quality isolates into IMG. For that, we first compute some basic statistics and then make sure that the coding density is at least 70%, and additionally that there are no intergenic regions longer than 3,000 base pairs. In any of these two cases, the submission would get put on hold and an email would get sent out to our in-house isolate experts. They would then look at the generated data and statistics and figure out why these quality metrics were not met. If it's just due to poor input data quality, for example, they would cancel the submission. If it's on the other hand due to a mistake during the feature prediction phase because, for example, a different translation table should have been used, then they report this information back and the submission would get reset and reprocessed with the correct translation table. Another important step that happens during the QA/QC phase is the flagging of the data set as either being of high or of low quality. This flag later determines if an isolate will end up in IMG-NR's NR database or not. The NR database gets built from all public high quality genomes in IMG and is used during the functional annotation phase, as I will show you on the next slide in a minute. If any of the following rules is true, the genome will get marked as being of too low quality to get used in our IMG-NR database. So max and unscreened single cells will automatically get blocked from ending up in there. And if the GOLD phylogeny is unclassified, the isolate can't get used for it either. Also, if the coding density is below 70% or the number of genes per million base pairs is either below 700 or above 1400, or if the number of sequences per million base pairs is above 300, the genome will not end up in our IMG-NR database. And after this, the final phase of the pipeline gets kicked off, which is the functional annotation. Now, for this, we take the predicted protein sequences and first compare them via last against our just mentioned IMG-NR database to get their KO, EC, and phylogenetic assignments. This is followed by several HMM search runs against the TigerFam, PFam, Cox, Smart, Cath, FunFam, and last but not least, the superfamily databases. For isolates, we also predict signal peptides via signal P and transmembrane helices via decode and HMM from the TM-HMM package. And in the final step, we then assign product names to the CDS genes. For this, we check if the protein has either KO or TigerFam or COC or PFAM hit, which we check in that particular order, and if so, transfer its product name. If not, we assign the term hypothetical protein. In the end, we then have the final nucleotide and protein FASTA files and the GFF file containing all predicted features and their functional annotation, as well as specific files from each module that list things like alignment coordinates, e-values, bit scores, and such. All these files get then handed over to the IMG database folks who start the process of integrating the data into the system for your further analysis. All right. And before I finish now and open the floor to questions again, I quickly want to show you guys what your data set looks like after it got annotated and loaded into IMG. And for that, I will switch back to my browser. All right. So this is the genome details page, and it lists the Clavia-Bacta genome. We actually imported it recently from GenBank and ran it through our annotation pipeline. So what you see here in the beginning, you first see a little bit information about your genome plus the metadata we pulled out of GOLD. And you also see here the pipeline version that was used to annotate your genome. And if you click on the plus before it, it even lists all the tools and databases and the specific versions that were used for it. After the general overview, it shows you the genome statistics that lists, for example, all the different gene types that got predicted or how many hits were found to the specific databases. All these numbers are clickable, of course, and would bring you to other pages with more details to explore. And towards the end, you will find even more tools to lure you deeper into the rabbit hole. And a lot of these tools and statistics will be covered in upcoming webinars. And to come full circle, the page actually also provides a link back to the original submission. And here you can see all the files that got created by the pipeline and the different phases and states the pipeline went through and how long each step take. And if you click on show statistics, it can also provide you with some basic feature statistics already right after the pipeline ran and before the data set got loaded into IMG. All right. Okay, back to the IMG page. And last but not least, I should also mention that there's extensive documentation on the IMG site that's accessible via the help menu entry. Here you can find, for example, documentation on the annotation pipeline, and then IMG and even IMG submission FAQs page. So if you have any questions, I would encourage you to first have a look here. You might save yourself some time since others might have the same question and we answered it already. If you don't find an answer to your question there, though, then you can, of course, always file a ticket with us via the contact us link and ask us directly. Well, and with that, I will turn to Natalia and Rika again to see if there were any general questions in the chat in regards to the submission or the annotation pipeline overview I presented that we should mention and comment on before we end the webinar.
We have one question that just showed up. It said, if you have a study where you want to annotate genomes of several isolates, do you have to submit individual sequencing projects? Do you have to submit them all individually? Can you do it in bulk? I guess this is a question for Reddy.
Yes, those sequencing projects, yes, has to be submitted individually because each genome is different, right? They all can go under one study, but projects has to be different. They will have their own analysis projects too. And if entering into the GOLD, if there are like more than a dozen or so, we can provide a batch load Excel and help the users to define the things in GOLD in a batch. Is that?
Yes.
All right. Yeah, and as for the actual data set submission, unfortunately, there's not an option yet to do batch submission. So you would have to go through the process of, submitting each data set individually. If you, though, have basically hundreds of them that you want to submit, you can always send us an email and provide us a file that lists all the AP IDs with the corresponding, let's say, URLs where you put your data, and we can create those submissions for you. All right. Are there any more questions?
Nope, that's it. Thank you.
Then I guess I should point out again that there's a new series of webinars starting soon and that the registrations for it just opened. And yeah, thank everyone for joining. Hope you got something out of it and will join us for any of the following webinars again. Thank you and bye everyone.